Hello, welcome to Biochemistry Simplified. Um, Good day, everyone. We are going to continue from where we left off now, and in continuation of where we left off, we're going to start talking about the nucleotides and the nucleosides of nucleotides and their sugars. The last place where we left off is that we started talking about that apart from the bases, the DNA molecules are made up of sugar phosphate backbone, as shown in this diagram. Okay, so there are a little bit of corrections I want to make. This is a moiety right here. Let me do this in red. All right, doing this in red. This is a moiety of the one of the nucleotide. Okay, the whole of this is a nucleotide, and this could represent the pyrimidine. This is a base. This is the base here, right here, a base, and this could represent what? The pyrimidine, which is a nucleotide. Pyrimidine. Why right here you have another base right here, and of course this is a base, and this in itself could represent a purine. So again, this is also a nucleotide. So these nucleotides and these nucleotides are building blocks of what? DNA molecule, and they are bases that are made up of sugar phosphate backbone, and this sugar phosphate backbone is represented right there. Excuse me. So we just move right along. In our next set of lectures, we begin to look at the three-dimensional structure of the DNA molecule. On the next page, please. We begin to look at the 3D structure, which is the three-dimensional structure. The three-dimensional structure of a DNA molecule. So three-dimensional structure of a DNA molecule. Alright, so the three-dimensional structure of a DNA molecule actually consists of two long helical strands that are coiled around a common axis, forming a what? A double helix. So you have the three-dimensional structure forming a double helix like this. So let me do this in maybe in blue. So you have a DNA molecule. Okay, and you have its backbone. Sorry about that. Okay, so this is it, but I could do better than this, so let me just take the diagram again. Sorry about my pen. Okay. Okay, so you have the three-dimensional structure looking like this, and you have the bases. So I'll just point out where the bases are situated in the three-dimensional structure. So you have things like this representing the bases. So that is it. Okay, so that's basically it. So you have the completion of the base structure this way. Alright, so this backbone here is referred to as the sugar phosphate backbone that we saw earlier structurally. So this is the sugar phosphate backbone. Alright, then these internal bonds are referred to as the base pairs. The base pairs. And of course, you have specific nucleotides that are bonded A to T, that's adenine, to thymine. Then you have G to C, which is your guanine to cytosine. Like you can see here, guanine for G this way and cytosine. Okay, so those are the base pair like we earlier saw in the structure of the base pairing of nucleotides in DNA molecule. So this basically it, and we could take down the following: that the three-dimensional structure of DNA consists of two long helical strands that are coiled around a common axis. Great. And this coiling around a common axis is actually what forms the double helix of DNA structure. Okay, so it's a forming a double helix Alright, so DNA is actually packaged this way. Okay, and they are packaged in chromosome pairs, which are the strands of what? Genetic material. They are packaged in chromosome pairs, which are the strands of genetic material, one coming from each parent. So we write here DNA is packaged in chromosome pairs, which are the strands of genetic material, one coming from each parent. So one of each strands, okay, one coming from each parent. One of each strands coming from each parent. Okay, so that's pretty much it. I would like to quickly give a key right here to indicate what is guanine and what is adenine in the structure above. So you have a key like W. W stands for what? Your cytosine. And you have the red. The red stands for your guanine. And you have the T. It's actually just two colors. So you have the AAT, okay, standing for the blue again. So maybe let's say purple, standing for thymine. And green, standing for adenine. So I'll just correct that in the diagram here. Okay, so this half is green and you have your purple right here. Okay, so while you're red, good. So that's a short key for the structure. Alright, so we now go ahead to look at how this DNA is packaged in chromosomes. Okay, so you have like a chromosome strands, and we could see that in the next page. So you have like a chromosome strands that has the gene segments, and the gene is called this way. Alright, so these chromosome strands could be thicker, something like this, so that these segments, let me do that in red, so that from this segment to this segment you have represents where you have the gene, and the strands can contain a helix that forms down, forming like a butterfly, and this actually represents the chromosome. Okay, so we said that the genes which controls an organism's characteristics are located in specific segments of these chromosomes. All the genes carried by a single gamete, that is, by a single representative of each of all chromosome pairs, is known as the genome, and the total genome size differ considerably in different organisms, as we can see subsequently. So this is like a structure of what the genes that are made up of DNA, and showing clearly the chromosomes. So if we note here now, we'll say the genes The genes which control the genes which controls an organism's characteristics are located on specific segments of each chromosome. All of the genes carried by a single gamete is known as what? A genome. Okay? All of the genes carried by a single gamete is known as a genome. Alright, and this single gamete, it represents a single representative of each of all of the chromosome pairs. So the single gamete in question, that is by a single, is a representative of each of all chromosome pairs. Okay, so that's basically it, and these have considerable different sizes. Okay, the total gamete sizes actually differs from each other. The total gamete sizes differs from each other, as you can see in the table below. So I'll do that in the table below. The total gamete sizes, let's note that the total gamete, sorry, I mean to say the total genome sizes. Okay, so let me just take that off. The total genome sizes differs quite considerably in different organisms. They differ quite considerably in different organisms. So, as shown in the table below, Okay, so I'll just go ahead and show you the table of different organisms having different genome sizes. So let me do this maybe in green. So in this table we have something like the organism as a field. Then we have the number of chromosomes. Then we also have the genome size, in base pair. Sorry, it's too close. Okay, so we have the genome size in what? Sorry, base pairs. Okay, so in a tabular form, we'll just quickly look at this. Alright, so if we take the organism bacteria, we say that bacteria has just one chromosome. Okay, that means it's a unicellular organism, and it has approximately 4,000 to 10,000, 400,000 actually, 400,000 to approximately 10 million base pairs. Okay, then you have the yeast, Saccharomyces cerevisiae, having 12 chromosomes, and it has 14,000 or 14 million base pairs. Then we also have the worm, and the worm has 6 chromosomes, and that is approximately 100 million base pairs. Then we also have the fly. We also have the weeds extending our table, and of course we have human. Now the fly has 4 chromosomes, the weed has 5 chromosomes, and the human has, as you all know, 23 chromosomes. Now the fly has 300 million base pairs, 300 million base pairs. Then you have the worm having 125 million base pairs, and of course the human has up to or more 3 billion base pairs. Okay, so note that these sizes determine the upper bound of genetic information in the organism. That's very, very key. Now, the genome sequence is now available in direct method to help identify specific genes. An indirect method of achieving the same aim is through the use of what? Genetic markers. Okay, so we'll move right along to knowing what genetic markers are and how they can indirectly determine how they can easily determine the indirect method of achieving the specific, of identifying the specific genes. So want to write here notes. So we said genome sequence. Allows for the tracing of a specific region, tracing of a specific region of DNA, or as a specific piece of DNA, or as a specific piece of DNA with a known with a known position on the genome. With a known position on the genome. Okay, great. So that's two key definition of genetic marker. It can be defined as a gene whose phenotypic expression is usually designed or determined and is used to identify an individual or a cell that carries it. It could also be defined as what? A chromosomal landmark or allele that allows for the tracing of specific region or DNA, or as a specific piece of DNA with a position in the genome. Of what importance is this? All of these are important for the genetic engineering process. In our next slide, we'll begin to look at the genetic engineering as a subtopic and all other terminologies around it. Next slide. Okay, so we have genetic engineering, also known as not just genetic engineering, we have genetic engineering, recombinant DNA technology, genetic modification and manipulation, and gene splicing are all terms that apply to the direct manipulation of an organism genes. So we look at genetic engineering, recombinant DNA technology, okay, genetic modification, or manipulation, or in abbreviated as GM, and genetic splicing or gene splicing. Gene splicing are all terms, they are terms that apply to the direct manipulation of an organism's gene, of an organism's genes. Okay, so how does this happen? Genetic engineering uses the techniques of molecular cloning and transformation. Genetic engineering uses the technique okay, it uses the techniques of molecular cloning right? Uses the techniques of molecular cloning and transformation. Okay, techniques of molecular cloning and transformation to alter the structure and characteristics of genes directly, and characteristics of genes directly. So these are the techniques it uses of molecular cloning and what transformation to alter the structure and characteristics of genes directly. How can it be applied? Okay, genetic engineering can be applied or have been applied, have been shown to be useful in various what industries, principally for medicine and what agriculture, with some recorded success. So we see the application of genetic engineering. We say the genetic engineering techniques, genetic engineering techniques have been applied they have been applied to various industries. They have been applied to various industries. Applied to various industries. Principal of them are medicine and what? Agriculture. Principally medicine and agriculture. Alright, principally medicine and agriculture, with some recorded success. That means genetic engineering with some recorded success. That means the techniques of genetic engineering has been applied and we have seen it to be successful in some areas, principally in the industries of medicine and what? Agriculture. So we see some examples in which genetic engineering have been applied. So we say examples include, let me do examples in another color, say blue. So we say examples of genetic engineering application. So examples include the following. Examples include the following. One on the list is the production of synthetic human insulin. Include the following. One is the production of synthetic human insulin. The production of synthetic human insulin using modified bacteria. Using modified bacteria. Okay, that is one clear example. We have example number 2. Okay, and example number 2 is the production of erythropoietin. The production of erythropoietin. Alright, production of erythropoietin in hamster ovary cells. In hamster ovary cells. In hamster ovary cells. Then we also have the production of new types of experimental mice. The production of new types of experimental mice okay, such as the oncomice, the oncomouse. Onco simply means cancer cells, so oncomouse can also be called cancer mouse. The oncomouse, okay, for research purposes, okay. And four, we also have the production of new strains of crop plants, okay, the production of new strains of crop plants with beneficial qualities, all right. So these are examples in which genetic engineering has been applied in the various industry. As we can see, even from our example, it has been principally applied to the industry of medicine and agriculture. However, it is worthy of note that the first step involved in genetic engineering manipulation or modification is the isolation of a DNA molecule. So if you must go the genetic engineering way, you must understand that you have to isolate the DNA molecule of whatever organism your research is based on. So we'll continue on the next lecture on the isolation of DNA. See you in the next class. Thank you for joining Dr. Elick today at Biochemistry Simplified.